The next phase is metaphase 1, which is where spindle microtubules line up homologous pairs in the center, and random orientation occurs of these pairs. Random orientation is the random lining up of homologous chromosomes in the middle of the cell. And from these homologous chromosomes, there are chiasmata, which is a point where a pair of chromosomes remain in contact.
The next phase is anaphase 1. So anaphase 1 is when homologous pairs are separated, and they're basically being pulled apart to opposite poles. One chromosome moves to each pole. And ambivalent is a pair of homologous chromosomes held together by at least one DNA crossover.
Then the next phase is telophase 1, which is where the cell is divided across the equator and chromosomes uncoil, and this is where diploid to haploid is complete.
So next we're moving into the second set of phases. So the first stage is prophase 2. And in prophase 2, the chromosomes coil up again, centrioles move to cell poles, and the nuclear membrane disintegrates. So these disintegrate.
Interesting. So next is metaphase 2, and this is where spindle fibers attach to the centromeres, and chromosomes align along the outer equator. Okay.
Next is anaphase 2. Anaphase 2 is when the spindle fibers shorten, centromeres split, and chromatids of each chromosome travel to opposite poles.
The camera's shaking. Alright, so next is telophase 2, and this is where the nuclear membrane forms around chromatids at each pole. Both undergo cytokinesis, chromosomes uncoil, and a nucleola
So also is cytokinesis. We're just gonna outline what that is for you because it happens in both telophase 1 and telophase 2.